So I'm going to describe the development of Timd2 as a reporter gene for magnetic resonance imaging, which we've demonstrated both in vitro and in vivo. This is work that was done by Stephen Patrick while he was a PhD student in my lab. So the development of gene reporters for MRI has a long history. Perhaps two of the most notable papers were published in 2000, the first from Ralph Weissleder's lab, where they used the transferrin receptor as a gene reporter. So expression of the transferrin receptor on the surfaces of the cell as a reporter. They then detected expression by using transferrin conjugated to iron oxide nanoparticles. So they would bind to the receptor, taken up into the cell, and then you could detect that in T2 star-weighted images as negative contrast. Also in 2000, Scott Fraser, Tom Mead published another reporter based on beta-galactosidase. Now, beta-galactosidase has been widely used as a gene reporter for in microscopy, for example. They designed a gadolinium chelate in which the gadolinium was partially shielded by a galactosyl residue. Cleavage of that galactosyl residue exposes the gadolinium to water, and you can detect that in T1 weighted images, and you can see an increase in signal intensity. Subsequently, another well-known reporter, quite relatively widely used, I guess now, is ferritin. So ferritin, as we'll see later, is an iron storage protein. Will give negative contrast in T2 star-weighted images. So you express ferritin as a reporter and then detect the presence of the reporter expression in T2 star-weighted images. More recently, another interesting example from Jeff Bulter and Peter Van Zyl is the lysine-rich protein, an artificial protein containing many lysine residues. And they detected expression of this reporter using CES by looking at chemical exchange saturation transfer between the amide protons on LRP and water. And then two much more recent examples from my lab. The first published in 2013. This is using OATP. OATP is predominantly a liver membrane transporter. It's fairly promiscuous, transports quite a wide range of different molecules. It's responsible for the uptake of a hepatotrophic contrast agent, gadoxetate. So by using OATP as a reporter, when this is expressed, the cells will take up this gadolinium contrast agent, which then can be detected in T1 weighted images. And we showed several-fold increase in signal intensity in OATP expressing cells in vivo. So quite a sensitive reporter. It can also be used for other imaging modalities. So we showed if you switch out the gadolinium and put in a radionuclide, for example, then you can detect that using radionuclide imaging. We showed that with SPECT. And then remarkably, it turns out that OATP will transport luciferin, which is the substrate for luciferase, which we can detect with bioluminescence imaging. And we showed it could produce up to an order of magnitude increase in bioluminescence. So really a multimodality reporter. And then very recently we showed that you can use the urea transporter as an MR gene reporter. In this case, we detect reporter expression using hyperpolar IC13 labeled urea. And we detected uptake of the urea into UTP expressing cells using diffusion-weighted spectroscopy. So when the urea goes into the cell, you see a decrease in this apparent diffusion coefficient. So the first thing we did was to create a lentiviral vector carrying the Timd2 coding sequence. And we co-expressed Timd2 with M. strawberry, which is a red fluorescent protein. And we achieved co-expression by putting an E2A sequence between the coding sequences for M. strawberry and Timd2. So that ensures co-expression of the two proteins in nearly equimolar proportions. And we drove expression using the PGK promoter, which is a relatively strong promoter in mammalian cells. We then transduced, in this case, HEC293 T cells with this lentiviral vector. The cells expressing Timd2 would also express M. strawberry, and so we could detect whether Timd2 was being expressed by looking for the red fluorescence from M. strawberry. So we showed that Timd2 expressing cells would take up ferritin using initially fluorescence microscopy. So we used ferritin conjugated to FITC. FITC is a fluorescent dye, and by using fluorescence microscopy we could monitor the uptake of that labeled ferritin. Now, as I just mentioned, we can detect the presence of Timd2 expressing cells by looking for red fluorescence. So if you look at the bottom panel, RFP, the red fluorescence tells us that those cells are also expressing Timd2. When we incubate them at different ferritin concentrations over a period of 3 hours, we can see as we increase the ferritin concentration, we see increased uptake of the fluorescently labeled ferritin. And note that the control cells, i.e. not expressing Timd2, show no uptake. So in this experiment, we've looked at the uptake of manganese-loaded ferritin, and we can detect manganese-loaded ferritin. So in this, instead of loading ferritin with iron, we've loaded it with manganese, and we can detect that through an increase in spin lattice relaxation rate. And if you look at on the left panel, what you can see here are T1 weighted images of cell pellets on the left expressing Timd2 and on the right control cells, and we've incubated them with manganese-loaded ferritin. And as you can clearly see, there's an increase in image contrast in the Timd2 expressing cells, showing that they've taken up this manganese-loaded ferritin, and we can detect that in these T1 weighted images. And if you make measurements of the relaxation rate, you can see there's a really quite significant increase in those cells that have been incubated with the manganese-loaded ferritin. There's an increase in R1. Instead of using manganese-loaded ferritin, now we've used iron-loaded ferritin, so we get negative contrast in T2 weighted images. And you can what you see here are cell pellets from Timd2 expressing cells and control cells. From the control cells, no uptake of the iron-loaded ferritin. In these T2 weighted images, the pellets are bright. And conversely, in the Timd2 expressing cells, which have taken up the iron-loaded ferritin, you see negative contrast, you see a decrease in signal intensity. And if you look at the tubes in the bottom panel, you can clearly see in the Timd2 expressing cells that have taken up ferritin, you can see a color change in the cells because there's significant iron uptake. In this experiment, we've correlated increases in R2 relaxation rate with increases in cellular iron content. So if we just look at the Western blot shown at the bottom, alpha-tubulin is just used as a control for loading. If you look at the Timd2 expressing cells, when we blotted for ferritin, an antibody against the L chain of ferritin, then you can clearly see over a period of time that there's an increase in ferritin content in the Timd2 expressing cells, whereas there's no uptake in the control cells. Now, if you look at the top left-hand graph, what we've plotted here is R2 spin-spin relaxation rate versus incubation time. In the Timd2 expressing cells, we see an increase in R2 over 60 odd hours or so, whereas there's no uptake in the control cells. If we look at iron content, so the increase in R2 is paralleled by the increase in cellular iron content, whereas if we look at the control cells, there's really no significant change in iron content. So we didn't take manganese-loaded ferritin forward for studies in vivo because it became quite clear that there was some toxicity associated with the manganese-loaded ferritins. So if we look on the graph on the left-hand side where we looked at cell viability, you can clearly see that in Timd2 expressing cells incubated with manganese-loaded ferritin, there is some loss of viability, whereas in controls, one doesn't see that. And if you look at the right-hand side, where we're looking at cell growth rate, in cells incubated, in Timd2 expressing cells incubated with manganese-loaded ferritin, there's quite clearly an inhibition of cell growth. And we don't see that in control cells expressing incubated with manganese-loaded ferritin, or Timd2 expressing cells incubated with iron-loaded ferritin. So that's really quite an important point. Iron-loaded ferritin has no effect on cell growth rate, but clearly manganese-loaded ferritin does. And therefore we took ferritin forward as the contrast agent that we would use in vivo. So this is the final experiment which we did in vivo. So we've injected Timd2 expressing cells on one flank of the animal and control cells on the contralateral flank. We then injected intravenously iron-loaded ferritin and looked at the development of image contrast over a period of 48 hours. And after 24 hours, we can clearly see in the Timd2 expressing xenografts there is an increase in R2, and that's significant when compared to control cells on the contralateral flank. We clearly don't see as big a change in R2 as we saw in vitro, and there are a number of possible explanations for that. For example, ferritin may have limited access to the tissue. Ferritin has a relatively short blood half-life. We think possibly we could increase image contrast, for example, by using multiple ferritin injections and effectively loading the cells over a period of time. Nevertheless, clearly there is, we have observed image contrast in vivo. So in summary, we've demonstrated that Timd2, this membrane receptor which transports ferritin, can be used as a reporter gene for MRI. We showed in vitro there were really quite large increases in R2 and R1 when we incubated cells with ferritin loaded with iron and manganese loaded ferritin respectively. We didn't pursue manganese loaded ferritin in vivo because there was some evidence of toxicity associated with this agent. The contrast that was produced in vivo was much less than we saw in vitro. A number of possible explanations for that, for example, fast clearance of ferritin from the circulation, and possibly ferritin is quite large and so poor delivery to the tissue. And we suggest that potentially sequential injections of ferritin may be used to improve image contrast. Recent work from our lab where we've really concentrated using small molecule agents, small molecule contrast agents. So for example, we showed with OATP, we're using a gadolinium chelate gadoxetate, which gives positive contrast in T1 weighted images, and we also have shown that urea can be used, hyperpolarized C13 labeled urea can be used when we use the urea transporter as the gene reporter. And I think the advantage of these reporters is the contrast agent is much smaller and therefore should have better access to the tissue.